Okay, good afternoon, class. Today, in this video lecture, we'll try to cover your major histocompatibility complex. And if you try to browse your reference books, events, you'll notice that this topic is a subtopic under your antigen. And in the previous video lecture, we're able to cover your antigen already. So, to complete the interaction of your innate immunity to your adaptive immunity, we'll try to fill the gap by discussing this topic, major histocompatibility complex. Again, I am your professor for this video lecture. I am Alfredo Ine Jr. If you have some questions and or clarifications, please don't hesitate to contact your professor, and I will try to clear the gray areas of your immunology and serology. Okay, so let's start the discussion. So in this video lecture, you can follow using your reference book, Stevens, and with your secondary reference book, which is Sturgeon. This topic is very short only, so please bear with my video lecture. Okay, at the end of the course or topic, you are expected to at least able to discuss the major histocompatibility complex or MHC from a historical perspective. So your MHC, you also have another term which is your HLA or human leukocyte antigen. So they're interchangeably used in the context of your immunology and serology. And I'll be discussing shortly. I'll just put some inputs of the MHCHLA in a historical perspective. Then second, list the classes of your MHC molecules with its corresponding form. So you have your class 1, 2, and 3, but we'll try only to discuss specifically your class 1 and your class 2. Why? Because these are the two classes that are very important in terms of your processing with intracellular and extracellular organisms that are discussed in our clinical bacteriology. And of course, in relation of our clinical bacteriology, we'll try to merge what you learn in your clinical bacteriology to your immunology and serology. So this will hit our objective number 3 and number 4, which is describe how exogenous antigens, those are extracellular organisms, are processed and displayed on a cell surface in association with your class 2, specifically the activation of which T cell, CD4 or CD8. And lastly, describe how endogenous antigens are processed and displayed on cell surface in association with your class 1. So basically, we'll only discuss the structure, function of your class 1 and class 2, and how does your class 1 and class 2 helps the interaction of your innate immunity to your adaptive immunity. Okay? So if you can observe here, this is a interaction, interaction of your innate and adaptive immunity. So we need to identify first class. Sir, saan yung innate dito? Saan din yung adaptive? Okay, so as we discussed in the previous video lecture class here, that your adaptive immunity, immune cells are composed of your B cells and your T cells, and also some reference book consider NK cell as part of the adaptive. Okay, so in the case here, so you have your T cells here, this one, T cells, B cells, and your plasma cells that will eventually produce antibodies. So this one is your adaptive immunity. And on this part, your antigen-presenting cells, you have your B cells. They are also considered antigen-presenting cells. Second is you have your macrophages. Okay, macrophages are the most potent antigen-presenting cells. So if you can observe here on the innate immunity part, so they will try to process that one. Okay, sir, bakit kailangan pa i-process yung antigen before i-present sa T cells? Can the T cells cannot recognize the antigen itself? To answer that one, class, tama. Yes, correct. Okay, your T cells does not have any capability to recognize the whole kay antigen. So ano yung gusto niya lang class? Mapili kasi si T cells, yung gusto niya lang is yung mga antigen na na-process na. Okay, so they cannot process the whole entire organism. For in the case of Staphylococcus aureus, they cannot identify and recognize that one unless the antigen presenting cell will try to engulf that one, process that one, and present using your class 1 and your class 2. So it means that your MHC or major histocompatibility complex is the bridge. Okay, it's the bridge between your innate immunity to your adaptive immunity class. Okay, so take note with that one. Ganito ka-importante si class 1 and class 2 in terms of presenting the antigen okay, recognized on the innate part to your adaptive. Okay, so as mentioned in the slide, their main function is to bring antigen to the cell surface for recognition by your T cells because T cell activation will occur only when antigen, okay, take note with this one class, when processed antigen is combined with your class 1 and or your class 2 molecules. See here, para you have a good picture of how your innate and your adaptive immunity will interact. So have your biological compounds in the case of bacteria. You have here, so for example, this one is your Staphylococcus aureus. And your Staphylococcus aureus, of course, will be engulfed by your antigen-presenting cells. In the case of the most potent antigen-presenting cells, you have your macrophage. Then they will try to process that one. They only select the small peptide, not all, and then present that one using your class 1 and or your class 2. Eh, and then after that one class, they will your CD4 T cells will be activated. Either they will produce cytokines. We will try to discuss that one on a separate video lecture. And then now your activated B cells will also activate your activated CD4 T cells will also try to activate your B cells, and your B cells will mature to plasma B cells. Then plasma B cells will produce antibodies that will try to opsonize, neutralize your antigen. And fortunately, you were able to discuss and cover your antigen and your antibody characteristics in the previous video. So I hope that this video will try to, connect because I think this is our last topic with your innate and your adaptive. And after this one, we will jump off to your autoimmunity. Like what will happen if there are some malfunction under your innate and your adaptive, then we'll move to the diseases already. And so the purpose of this slide is for you to at least get a one picture from what we discussed on the innate down to your adaptive. So as presented by your reference book, we have two terms. Okay, so you have your major histocompatibility complex, and then you have your human leukocyte antigen. So in general, okay, these are the gene system which play a key immunologic role. As I mentioned earlier, this will bridge your innate or your adaptive by activating your adaptive immune cells, specifically your B cells and your T cells. Okay, so in humans, they are found on your short arm of your chromosome 6, band 21. So here you have your chromosome 6, then short arm, then that's 6p, then 21. And then under this one, you have your major histocompatibility complex class 1, class 2, and class 3. So they encode for human leukocyte antigen or HLA for humans. So your human leukocyte antigen, okay, there are molecular basis for T cell discrimination to determine kung sino yung mga cell from your non-self antigen. So then therefore, class, these are markers, okay, markers that indicates that this part of, for example, cell or organ are part of the body. Okay, so this is the marker, a tag that this specific cell contains a specific antigen that is is self, not a non-self. Okay, so your HLA or human leukocyte antigen are named by a French scientist, Dosset. And then the main reason why they are called human leukocyte antigen is because the discovery it was first discovered on your leukocytes. The discovery of your antibody response to a circulating WBCs. Then this one they term okay human leukocyte antigen because there are some circulating antibodies of your circulating antibodies that will direct to a specific WBCs. Then these antigen are called human leukocyte antigen. Then they found out later na hindi lang pala leukocyte ang merong HLA. Then to clear this up, they use the additional term. You have your major histocompatibility complex. Okay, your MHCHLA is a gene system. Your major histocompatibility complex the term itself will be discussed on your transplantation. Kasi with the histocompatibility, it means that this is also will cover with your transplantation. Okay, so among all the system in our body class, this is the most polymorphic. Okay, to increase the chances of interacting with diverse self and non-self antigen or immunogens. The main reason why they are called polymorphic class will be explained on the previous slide because I have a separate slide with that one. Okay, so you can understand why they are called a polymorphic. Then your MHC class, MHC HLA gene system is divided into three categories or classes. So you have your class 1, class 2, and your class 3. So here, if you can observe, this is your HLA, MHC gene. Then you have your class 1, class 2, and you have your class 3. Red alert tayo class because these are the some questions that usually appear on your board exam and or pre-board exam. So in preparation for that one, we need to at least memorize this one. So for your class 2, is if you can observe here, your class 2, okay, located here, okay, your alpha chain and one or more genes that coded for the beta chain are located. Okay, so you have your alpha and you have your beta chain. Okay, your class 2 region. Okay, and then you have your class 3 here, which code for complementary proteins and cytokines, such as your TNF or tumor necrosis factor. So your class 2 here is located. Class 1, your class 3. Here. Okay, then your class 1 and your class 2 are gene products are involved in antigen recognition. This is the main reason why we will try to cover your class 1 and your class 2. Your class 3, as I mentioned earlier, here, this area or region is the main responsible of producing and encoding your complement proteins. And we discuss your complement proteins, right? You have your C1QRS for your classical pathway, then production of your C3 convertase to your MAC complex. So the MHC system or major histocompatibility complex system is described as polymorphic as I mentioned earlier, right? Because there are so many possible alleles at each location. So imagine class here on your class 1, okay, your HLA-A on your class 1 have 580 different alleles. And your HLA-B under your class 1 have 921 different alleles. And then your HLA-C under your class 1 have 312K alleles. So this is the main reason why they are called polymorph because with the combination, sabi pa ni Stevens class, very less likely na na same yung combination ng mga alleles. And therefore, this will serve as the main reason why they are called polymorphic. Okay? Now we'll move to your structure function of your class 1, okay, versus your class 2. And we will try to cover first your class 1, then we'll move to your class 2. For the preparation of your assessments, exam, pre-board exam, and board exam, okay, majority of the questions are more on the structure of your major histocompatibility complex. So I want you to at least understand by heart, class. Okay? The main reason why we need to understand this one is not purely by preparation for board exam, of course, to understand holistically, okay, the beauty of your immunology and serology class. Okay. Okay, let's move to your MHC class 1. So you have your HLA1, HLA-A, B, and you have your C. So this is the structure of your class 1. If you can observe, it composed of the alpha, beta, of course. And then your alpha have three segments. You have your alpha 1, alpha 2, and then you have your alpha 3. Then you have your beta 2 microglobulin. Okay, your class 1, okay, MHC, uh, is expressed by all nucleated cells. So I remember during my college days class, my teacher usually asked me, even during the review, uh, this question. So, expressed by all nucleated cells. So, yung tanong usually lumalabas sa hematology class is the RBC class case. The RBC uh, have your class 1 expression on the cell membrane. So we knew already that your RBC is unnucleated. So they don't have nucleus, right? So majority of the students will try to answer, no sir, they don't have class 1. Kasi as we discussed during our hematology, with the RBC maturation series. At the end of the maturation, the RBC does not have any nucleus. Then therefore, dapat wala silang class 1, sir. But if you will try to go back to the start of your RBC maturation series, you can observe that the retrocyte in the first stage, they have nucleus. Then therefore, meron ng expression of your class 1. So to answer that one, class, either it will appear on your board exam, either in your immunology and or your hematology, if they will ask if your RBC has your class 1 or a class 1 expression, or for example, uh, in the board exam, they will try to ask that your RBC uh, contains the following major histocompatibility complex. A, MHC class 1, then class 2, then C, class 3, then D, none of the above. Majority of the student, of course, will try to answer D because they will try to think, RBC unnucleated. And therefore, wala silang class 1. But remember the RBC maturation series that on the first stage of your RBC, they are nucleated. And therefore, they have class 1 class in their cell membrane. Okay, take note with that one. So they are highly found on your lymphocytes, okay, low or undetectable on your hepatocytes, okay, muscle cells, okay, sperm cells, then you have your neural cells. Okay, take note with the following cells na present yung abundantly present yung class 1. And then your HLA-C are expressed the lowest. Then among the three under your class 1, your HLA-A and B are the most important. So here, if you can observe, you have your alpha 1, as I mentioned earlier, you have your alpha, and then you have your beta. These are two non-covalently bonded polypeptide chain. Okay, so let's start with your alpha chain. Okay, the molecular weight is around 45,000, kilodalton. And then folded into three domains. You have your alpha 1, you have your alpha 2, then you have your alpha 3. Okay, so polypeptide chain anchored to E cell membrane. So take note with this one, class, your alpha chain, alpha 1, 2, and 3, they are anchored to the cell membrane here. Your beta, 2 microglobulin are floating. Okay, so later I will explain the main function of your beta 2 microglobulin. So of course, the main function of your alpha chains is that your alpha 1 and your alpha 2 is the peptide binding cleave. The main function of your alpha 3 will serve as E anchor. And then here, specifically, we'll try to cover and discuss your alpha 1 and alpha 2 function. So your alpha 1 and alpha 2 will serve as the antigen binding site. And then will able to hold around 8 to 10 amino acids. So very small. Okay, so it should be processed first. Okay, before they will be handled by your class 1. Kasi tignan nyo naman class, very short yung amino acid niya. Okay, so 8 to 10 amino acid lang siya. And then, take note also with this one class because aside from your alpha 3, the main the function of alpha 3 is to anchor to your cell membrane. Other function of your alpha 3 is this part is detected with your CD8 or T killer cells. Okay, take note with that one. Okay, CD8 T killer cells or cytotoxic T cells. Then, let's move to your beta-2 microglobulin class, beta-2 microglobulin. So, the molecular weight is 12,000 kilodalton. Compare kanina with your alpha-1, 2, and 3 or alpha chains are 45,000 kilodaltons kasi malaki sila class. They are composed of 3. Okay, 1, 2, and 3. Alpha-1, 1, 2, and 3. But with your beta, they are smaller compared with your alpha because isa lang siya class. Okay, so you have your 12,000 kilodalton. Majority of the board exam also class, or I think 3 or 4 times I encountered during the review that the beta 2 microglobulin is encoded in what chromosome. That question usually appear. So what you need to do is at least memorize this one. Okay, so your beta 2 microglobulin is coded on your chromosome 15. Okay, and then the main function of your beta 2 microglobulin is to stabilize, okay, the folded alpha chain. Okay, kasi pag class, pag alpha lang, yan, tapos may space dito, hindi siya stable. Then therefore, the main function of your beta 2 is to stabilize your alpha. And so dapat may taga-support talaga class. Then with that one, you need to, in applying that one in a real life, even with your class 1 class, your alpha cannot stand alone. They need a supporter. Then therefore class, you need to find in your life your beta 2 macroglobulin. If you are class 1, you need to identify now, okay, is this person is a beta 2 macroglobulin or not? Will this person support me throughout or not? If not, then, leave that toxic, okay, person in your life so you can survive the course. Yan, okay? So, very, simple application of your class 1 and your class 2 in real life. Okay? So now, let's move to the antigen processing and presentation of your class 1. Okay, this part, I'll try to explain naman class kung paano nake-create, paano sinisynthesize ng antigen presenting cell yung class 1 natin. Okay? And then kung paano ba kinakapture yung class 1 from your antigen presenting cells to your adaptive immune cells, specifically your T cells. Okay? So, of course, with your class 1, it will always start with your intracellular pathogen. Okay? Intracellular pathogen, pwede ba tayong magbigay class ng mga example na intracellular pathogen na natutunan natin sa clinical bacteriology? Correct, di ba? You have your Neisseria, gonorrhea, Neisseria meningitidis. Those are intracellular organisms. So if you can observe on the slide, diba? Then you have your neutrophil. They're located inside. You have your Mycobacterium tuberculosis. Correct, they're intracellular class. And then also you have your Listeria monocytogenes. Those are organisms that can survive inside the cell. And then I hope that your clinical bacteriology will maybe not in a molecular level explain why these organisms are able to survive inside, but at least can be introduced the concept class. So going back to immunology class, so you have your intracellular pathogen. And then in your intracellular pathogen, of course, this will be inside the cell. For example, antigen presenting cell. So since they are intracellular intracellular pathogen, then therefore they are able to multiply, okay, inside the antigen-presenting cells. So for example, Mycobacterium tuberculosis, as I mentioned earlier, and other organisms. Okay? So during the multiplication of your pathogen inside your antigen-presenting cell class, uh, ang kagandahan ng mga antigen-presenting cells class, even that they allow the bacteria to reproduce inside, meron silang checking mechanism. Kasi during the multiplication of your pathogen class, okay, so they will try to allow, sige, replicate ka lang dyan, sabi ng antigen-presenting cell. Pero during the multiplication of pathogens, they also produce DRIPS class or defective ribosomal products. Okay, so during the, para siyang anong class, during the replication, meron talagang mga parts na metabolites ng mga organisms. So we call that one DRIPS or defective ribosomal product. And these defective ribosomal products class are very crucial in the production and synthesis of your class 1. Why? Because in the interaction of your proteosomes, okay, as we discussed this one already, right? I hope that this is also discussed in your clinical bacteriology. With your proteosomes, or in our phagocytosis, right, your proteosomes will try to degrade, okay, your drips into smaller part, okay, around 8 to 12 okay, amino acids. Okay, the main function of your proteasome is to cleave the drips into a smallest portion, around 8 to 12 amino acid. You need to memorize the amino acid class. Why? Because this is very important, specifically in your exam, in your assessment, and probably in your pre-board exam. So if the proteasome, sorry class, if the proteasome is able to degrade your drips into smaller peptides, then these smaller peptides now will go to your endoplasmic reticulum. So sir, paano ba sila pumupunta dun? Diba? They are inside the cytoplasm. So sino yung mag-guide sa kanila class? You have your top 1 and your top 2. Okay, your top stand for transporters associated with antigen presentation. So by the use of your top 1 and top 2, these are chaperone molecules. Okay, chaperone, ibig sabihin sila yung nag-guide ng antigen class to your endoplasmic reticulum. So inside the endoplasmic reticulum class, your your ribosome here, this one, your ribosome will try to synthesize now the first part of your class 1, which is your alpha chain. Okay? So ngayon, magpo-produce, magsisynthesize siya ng alpha chain, and then by the presence of your calnexin. Your calnexin class, the main function of your calnexin is to stabilize your alpha chain. 'Di ba? As I mentioned earlier, sino nga yung stabilize kay alpha? Correct. You have your beta, 'di ba? Beta to microglobulin, yun lang naman function ni beta, 'di ba? Kaso hindi pa gumagawa ng beta chain. Para maging stable si alpha calnexin because wala pa si beta 2. Okay, microglobulin. So siya muna yung mag-a-assist kay alpha chain. Okay? Tapos after nito, pag stable pag andyan na si calnexin, and then your ribosome will now produce your beta chain. Tapos with your beta chain, ayan, papasok na siya here to stabilize that's one. Tapos may additional na naman na mga molecules, calreticulin at saka ERP57. The main function of these two is, of course, to still support the alpha chain, and second, to cover the alpha 1, alpha 2. Kasi meron tayong mga endogenous class, endogenous antigen. Okay? Aside from the process 1, meron tayong mga endogenous antigen. Yung function ni calreticulin at saka ERP57 is to protect the binding site, na dapat yung binding site na alpha 1, alpha 2 will only bind to the pathogen peptides. Okay? Tapos, after nyan, papasok yung isang class, molecule that will also stabilize your class 1. Okay, you have your tapacin. And then, with this one, pag na-stabilize na ng tapacin, ma-remove na yung other molecules, then it will bind your antigen to your class 1, alpha 1 and alpha 2. Pag nag-buy na siya class, ready na siya to labas here. Dadaan siya with your Golgi apparatus. And then, pag dumaan na siya ng Golgi apparatus, ready na siya to present. Pag-present natin yan sa cell membrane here, of course, di ba, as I mentioned earlier, that the main function of your class 1 and class 2 major histocompatibility complex is to present the portion or process peptide from the antigen presenting cell to your adaptive immune cells. So with your class 1 naman class, this will interact to your CD8. Okay, interaction with your CD8 cells. Ito yung CD8 natin class. And then you have your alpha 1 and alpha 2. Okay, will present the antigen, and then your CD here class, your alpha 3 region of your class 1 will bind to your CD8. That's one. Okay? And then pag na-activate na, pag nag-interact na sila class, then your CD8 will release cytokines. Okay? Will release cytokines class. Yeah. Okay? So take note with that one. Okay. So that is the process of your class 1. Okay? So if you have some questions and clarifications, kapag hindi nyo pa naintindihan yung concept, please go back again class. Okay? So you can understand the process. Then let's move to your class 2 class. Okay, class 2. Okay, you have your class 2 major histocompatibility complex. This is the structure of your class 2 here. So it's still composed of your alpha in your beta. However, compare with your class 1, your alpha here in your class 2 is composed of alpha 1 and alpha 2. And then your beta is composed of your beta 1 and your beta 2. Okay, so this class 2 major histocompatibility complex is expressed by your antigen-presenting cells. Okay, macrophages, B cells, and then your HLA-DR is expressed at the highest level. So take note with that one. Accounts for about 1/2 of your class 2 molecules of a particular cell. And then your HLA-DR beta is the most highly polymorphic. So, of course, both your alpha and your beta are coded by your chromosome 6. Compare with your class 1, right? Because one of that one is encoded by your chromosome 15. Then both chains are anchored in the cell membrane. Okay, so you can now imagine, right? Class 1, isa alpha lang. Class 2, dalawa. So you have your alpha and your beta. And then, sorry class. Here, apologize for this one. I have a problem with the animation class. I'll try to fix it first. So, basically, that's the difference of your class 1 and your class 2 in terms of its structure class. Okay, so for your class 2, so your alpha and your beta is both anchored to the cell membrane. Okay, and then let's start with your alpha chain of your class 2. With the alpha chain, compare with your class 1, it's 45,000 because they have alpha 1, 2, then you have your alpha 3. But in your class 2, lesser compared with your class 1, 33,000 because dalawang subunit lang sa class. So you have alpha 1 and you have your alpha 2. Okay, both serves as a recognition site by your T helper cells by the CD4 markers class. Okay, CD4 markers. And then you have your alpha 1 and you have your beta 1 domain here. So we have your beta 1 domain here, class. Allow me to have my pointer. So you have your alpha 1 and you have your beta 1. Okay, domains. Okay, these are antigen peptide binding site. Compared with your class 1, meron tayong alpha 1, alpha 2. But with your class 2, you have your alpha 1 and beta 1. Okay, it can hold up to 13 to 17 amino acids. Compare with class 1, shorter, diba? Pagdating sa class 2, longer sa konti and up to 17 amino acids. Then the function of your beta chain is 27,000 kilodalton and folded of course with your beta 1 and your beta 2. So let's move to the antigen processing and presentation of your class 1 and your class 2 class. So compare with your class 1, we discuss more on the intracellular. Then therefore, the intracellular pathogen will try to go inside the APC. And then inside the APC, they will do the replication, correct? And then during the replication, they have a drips product. And those drips are degraded by your proteasome into a smaller peptides. Okay, so yun yung ano natin, process natin with your class 1. Okay, more on the intracellular organisms. But in your class 2, we are focused on your extracellular pathogen. So with your extracellular pathogen, as we covered this one already, with your phagocytosis, it will of course be engulfed with your antigen-presenting cells. And then during the phagocytosis video lecture, we were able to discuss that the engulfment or phagocytosis process will start with endocytosis. And then with your endocytosis, we'll have your endosome phagosome and fusion of your lysosome into phagolysosome. Correct? I hope that you still recall this one. Then due to the acidic environment of your lysosome, then your organism will be degraded into small peptides. And these small peptides will activate your endoplasmic reticulum, okay, by producing your alpha 1 and alpha 2. Okay, compare with your class 1, okay, I mentioned it earlier, right, that the production and synthesis of your alpha 1 and beta 2 are not simultaneous. Ibig sabihin, sa class 1, it will start producing first your alpha, then next is your beta. Okay, kaya meron tayong calcitine, diba? Calcitine ba yung class? You have your calnexin. Okay, you have your calnexin that will try to support your alpha chains. But in the case of your class 2, your ribosome will simultaneously produce your alpha and your beta chain. So, ito na yung itsura na class. So, you have your alpha 1, alpha 2, beta 1, and you have your beta 2. And then, in the process of meeting up your antigen, kasi di ba, ano pa sila, hindi pa sila nag-meet ni antigen class. So, dapat protected muna. Diba? Protected muna si class 2. So, sino yung magpro-protect sa kanila? Kasi you have your II antigens. Okay? You have your invariant chain antigens class, or we call that one II antigens. And then your II antigens will protect your alpha 1 and beta 1 because this is the antigen binding site of your class 2. Tapos, dadaan sila sa Golgi apparatus and then we'll have the transport. Then since due to the acidic environment of the cytoplasm, okay, so magkakaroon tayo ng degradation of your II antigen. And then, yung katawan lang ni II yung madedegrade class, yung ulo hindi. Okay, so you have this one. And then, we call this one as CLIP class, okay, or class 2 invariant chain peptide. Okay, so this one will still protect the antigen binding site, which is your alpha and your beta class, correct? So, siya pa rin yung nag-pro-protect. And then, in the presence of your HLA-DM class, this will, your clip will be removed. Then, ready na mag-meet. Ready na sila mag-meet class. Mag-meet na si class 2 at saka si antigen. Yan. So, pag nag-meet na sila class, tadan! Yan. Tapos, kukunin ni class 2 in the alpha 1, alpha 1, beta 1 antigen presenting. And then, we'll now process and present it to your, tadan! You have your CD4 naman class. Okay, you have your interaction with your CD4 T cells. Okay, as I mentioned earlier, both of your alpha will be detected with your CD4 T cells. Okay, compare with your class 1, diba, with your class 1, yung nadedetect lang is alpha 3. So, yung sa class 2, ang nadedetect is both your alpha 1 and your alpha 2. And then, with that one, your CD4 T cells will produce cytokines and then activate either your B cells and other T cells, okay, in other part of your innate and adaptive immunity. So here, if we can, so we are almost done, class. This is just a recap. Okay? So if you can observe your class, may mga palatandaan tayo, class, para hindi makalimutan kung ano yung itsura ni class 1 at saka ni class 2. Why? Kasi pagdating sa pre-board exam or with your exam assessments, there will be picture, then you need to identify if this type of molecule is a class 1 or class 2. So paano natin siya madedifferentiate? Madali lang, class. First is kung ilan yung naka-anchor. Okay, so if you can observe here, pag isa lang yung naka-anchor, class, madali lang yan, class 1. Okay? Pag dalawa yung naka-anchor, then therefore they are class 2. Okay? And then your alpha 1 and your alpha 2 will bind to your CD4, and then your alpha 3 will bind to your CD8. Okay? So you have your CD4, right? CD4. Sorry, class. So, may mga palatandaan ako nito, class. CD4. Tapos, kabaliktaran ng 4. So, ganito yung itsura niya, class. Kaya siya, helper. T cells ang tawag. Tapos, you have your CD8 naman, class. Here. Sorry. Erase ko muna, class, para maganda yung 8 natin. So, you have your sorry. You have your CD8. This one. Yan. Tapos, sorry, class, you have your CD8. Ito na lang. You have your CD8. Kasi kanina napakita ko naman yung CD4, diba? Tapos baliktarin mo lang yung 4 para maging H, kaya siya tinawag na helper cells. With CD8 naman, class, yung palatandaan ko dito before is, ayan, na erase yung lahat, class. Pag hinaf mo daw yung CD8, ayan, pag hinaf mo yan, class, ganito na yung itsura niya, CC. Kaya siya tinawag na cytotoxic cells. Yan. Para-paraan para hindi makalimutan, class. Okay, so you have your CD4 helper cells, then you have your CD8 cytotoxic T cells. And then, this one is a summary of your class 1 and class 2. Okay, with the synthesis of your class 1 and class 2 naman, class. Okay? So, ayun, that's it for your major histocompatibility complex or MHC. So, we can connect this one with your transplantation. You have a separate video lecture and discussion of your transplantation and how does your MHC HLA is very crucial pagdating sa transplantation. So, ayun, we're done. So, if you have some questions in or clarifications, class, please don't hesitate to contact your professors. And I hope that you understand the concept of your major histocompatibility complex. And ang gusto ko lang na message from this video lecture, class, is that your MHC will serve as a bridge. Okay? Kasi walang capacity si T cell to eat up, to recognize, kahit man lang ma-recognize, kasi wala silang capacity directly to recognize your antigen. So, mapili sila, class. Okay? Kailangan lutuin muna ng antigen-presenting cell, i-present sa kanila na this is antigen before they will try to react, which is very good. Diba? Kasi dapat, ma-process muna ng antigen-presenting cell yung foreign materials before mag-react si T cells and activate the other immune cells under your attack. Okay, so again, thank you very much, and I hope that you understand the concept of your major histocompatibility complex, and see you with the next video lecture, class. Okay, goodbye, thank you. Okay, goodbye and thank you, class. So I hope that you learned something. I'll try to stop. Bye, class. Bye. Bye, class.